Hello everyone, my name is Yash. This is my Journal Club 3 presentation. The article that I've chosen is titled The Impact of Delivery Mode on Colostrum Microbiota Composition. So let's start with the bottom line up front. The research question that was asked in this article was how does the difference in delivery method of neonates um, impact the microbiota composition in terms of bacterial abundance and the microbiota network. And so the study is comparing C-section delivery versus vaginal delivery. So why do we care? We care because colostrum or early breast milk is essential for the initial development of newborns. Essentially, it's a rich, nourishing substance that contains cytokines, antimicrobial peptides, mineral salts, antibodies, hormones, bioactive factors, etc. And so it's very important for a newborn child to receive these nutrients. So what's the need for this research? Understanding the differences can help to optimize neonatal health and potentially mitigate some of the risks that we know are associated with C-section deliveries. And so what did the authors find? Basically, they found that colostrum abundance and richness differs between C-section births and vaginal births. And they also found that the relationship between the microorganisms between these two birth methods differs significantly as well. And we'll get into that later on in the results. So for the background, breast milk is dynamic. So there's a difference between mature breast milk, and colostrum. Colostrum is early breast milk, so the first milk that lasts between 2 to 4 days after birth. And then it turns into transitional milk, and then at 14 days it turns into mature milk. And it's produced by the mammary glands. It's known to reduce the risk of gastrointestinal diseases. And so probiotic bacteria can actually reduce the chance of gastrointestinal disease by both competitive exclusion mechanisms and production of antimicrobial compounds. And so the colostrum provides up to 200 different species of microbiota for up to several months until after birth for the kid. And so it plays a very important role in immunity and metabolism. So how does the colostrum acquire microbiota in the first place? Currently, there are two accepted primary sources of breast milk microbiota. The first one being known as the enteromammary pathway. So if you take a look in the image, it's number 3. It's intestinal bacteria going directly into the mammary ducts by the means of dendritic cells and CD18 plus cells. The other way in which colostrum can acquire microbes is through something known as retrograde flow. It occurs during nursing, where the oral bacteria of the infants it could actually influence the colostrum microbiota during nursing. Now let's move on to talking about the sample, criteria, and controls of the study that was done. And so the study was done on 29 Italian mothers, 15 of which had vaginal deliveries and 14 of whom had C-section deliveries. So there were some specific exclusion criteria necessary. For example, they couldn't have been on immunosuppressive agents. They couldn't have been on antibiotic therapy. There shouldn't have been any significant maternal or infant illness, and there were no major birth defects. There were also some controls. And so all mothers had the same dietary intake. We know that diet can significantly impact microbiome composition. And so their diets consisted of primarily animal proteins, fats, and sugar. The samples of colostrum, so it was collected once, and it was collected up to 3 days postpartum. And when they were collecting the samples, the areola and its surrounding skin was disinfected. as well as the hand of the person who was collecting samples. Moving on to the methodology, once again, they collected one sample within 3 days postpartum. Uh, they did so after decontaminating the hand and the areola, and the sample was after it was frozen, it was thawed and centrifuged. After that, they did the bacterial DNA extraction, and so the total DNA was extracted, and they used something called a milk DNA extraction kit. This DNA extraction was then followed by a 16S gene sequencing, and so they used the 16S metagenomics kit to amplify the DNA, and then they used the ion torrent personal genome machine, or the PGM. And so how the PGM works is it measures the release of hydrogen ions during DNA synthesis. So as the nucleotides are incorporated into a growing DNA strand, protons are released, and this causes a change in pH. This change in pH is detected by a semiconductor sensor. And it's used to determine the sequence of nucleotides in the DNA sample, and it allows for high throughput and rapid genomic sequencing. And so after the gene sequencing, there was a statistical analysis was done. So there were 3 biodiversity indices, the Shannon index, the Simpson index, and the Chow index. And then they also set some, they also used the Kruskal, Wallace, and Mann-Whitney tests to set the to determine what counts as a significant difference. And then lastly, this is the interesting thing. I had never previously heard of this before, so I also had to learn about it. It was called the AutoCM map. Essentially, it's an artificial neural network. And how it works is that it finds the interactions between bacteria detected in C-section and vaginal delivery colostrum, and it does so by linking all the variables considered. And so it uses something called a minimum spanning tree algorithm, which maps only like the strongest correlations between system hubs that are connected to a node. And so essentially, it sounds complicated, but the results showed that bacterial hubs were basically the species that were predominant or most influential in a community. So it's useful because it highlights the significant role in health and disease for that bacteria. It allows us to see which bacteria drive community dynamics and the relationships that they have with each other. And so let's move on to the results. And so this is figure 1. It's showing bacterial diversity, and they're using a Shannon index, a Simpson index, and a Chow index to compare the C-section and vaginal delivery colostrum. And so it's important to recognize that there were no significant differences in biodiversity observed, even if the colostrum associated with natural delivery had a higher biodiversity compared to C-section colostrum. And so as you can see in the figure, both Shannon and Chow indices were slightly higher in natural delivery colostrum. So even though it wasn't found to be significant, it still shows that there was a higher biodiversity for natural or vaginal delivery. And so this is a crucial finding of the study because bacterial diversity promotes the maintenance and improvement of a balanced environment. So an ecosystem with a greater biodiversity, which is rich in a bacterial species, is most likely going to be more resilient and adaptable to stress than one with a low range of microorganisms. And so that's something to keep in mind as we move through the results of this study. So moving on to figure 2, we can see some of the bacterial differences in abundance that were found between C-section and vaginal delivery colostrum. We see that vaginal delivery was significantly richer in microorganisms belonging to Streptococcus and Haemophilus genera, they were more abundant by approximately 49% for the Streptococcus and 94% for the Haemophila. And then there was also a significant lower abundance of Finagoldia, Halomonas, Prevotella, Pseudomonas, and Staphylococcus for vaginal delivery compared to the C-section delivery. And so it's important to recognize that even though the vaginal delivery showed that it was more abundant in Streptococcus and Haemophilus, it's not necessarily a good thing. Because this can include both commensal and pathogenic microorganisms, so that's just something to keep in mind. Figure 3 has two parts, A and B. A shows the percentage distribution of aerobic and anaerobic bacteria in both C-section and vaginal colostrum, so it's both of them combined. And then B shows the relative abundance of aerobic and anaerobic bacteria in each of the colostrum samples. And so to characterize bacteria identified in the colostrum microbiota by sequencing, the authors used free online databases to discriminate between what they count as an anaerobe and as an aerobe, as well as to identify what they count as environmental organisms. And something important that I want to point out in this figure is that both C-section and vaginal delivery colostrum contained a higher number of anaerobic bacteria compared to aerobic bacteria. About 65% was anaerobic. Next figure, figure 4, it also has two parts, A and B. A talks about, A demonstrates the percentage of environmental bacteria in the colostrum samples, and then B demonstrates the percentage of intestinal bacteria in colostrum samples. So I think this figure is probably one of the most interesting figures of the whole study, because it shows that so part A of figure 4 shows that the colostrum of mothers who had a C-section was actually richer in environmental bacteria than mothers who underwent vaginal delivery. Now, this might be because of like during the C-section surgery, they had more exposure to environmental bacteria. And so that's very interesting to actually see that through this study. And then for part B, it's also interesting because both C-section and vaginal delivery colostrum showed a higher abundance of intestinal bacteria versus other bacteria. So it's important because this further supports the enteromammary pathway that we discussed earlier, where we said that one of the ways in which colostrum acquires its microbiota is through retrograde flow, and then the other way is through those dendritic cells and CD18 plus cells causing the intestinal microbiota to directly get to the mammary ducts. Moving on to figure 5. So this is the first of the maps generated by AutoCM, and it shows the microbiota network of C-section colostrum. And so the bacteria that they sequenced, they inputted it into the AutoCM artificial neural network, and that artificial intelligence generated this map of relationships between the bacteria in the colostrum for C-sections. And so the red circles, they represent all bacterial hubs that were identified. And so microorganisms that show at least 3 connections with other microbes in the network are considered as main bacterial hubs, right? And so as you can see, there are a ton of red circles, meaning a ton of bacterial hubs for the C-section colostrum, some of them including the Acromobacter, Delphtia, Enterobacter, Haemophilus, Rhizobium, etc., right? And so those were the bacterial hubs, and then there were things that they termed central nodes. And central nodes, there were 2 central nodes represented by Ruminococcus and Peptostreptococcus in the C-section colostrum, right? And why are the central nodes important? They're important because they give us insight into some of the key players of the C-section colostrum microbiota. So the two central nodes that were found, Ruminococcus and Peptostreptococcus, these are most likely critical to microbial community health and stability in C-section colostrum. And we should definitely further study their movement as the colostrum transitions and changes into mature milk. I think it would reveal a lot about uh, possible treatments and whatnot. Okay, moving on to Figure 6, which is the same map generated by AutoCM, except this time it's showing the network for the vaginal colostrum. And again, the red circles, they all represent bacterial hubs. And so the bacterial hubs were different this time. Some of them including, uh, you know, Causuria, Oscillobacter, um, Rallotella, etc. And so the two central nodes of this one, of the vaginal colostrum, were uh, Rhodanobacter and Acromobacter. And I want to point out that there were five nodes there were five hubs that were shared between the two colostrum microbiota networks. So for both C-section and vaginal delivery, there were only five bacteria that were shared. And now this suggests that these species play a significant role in the stability of colostrum microbiota, regardless of how uh, the child was delivered, and regardless of variations between networks. And their presence, their shared presence, highlights the importance of shaping the microbial environment of colostrum, and suggests that they might be key players in influencing early infant gut microbiome development. So as we move on to the discussion, I want to point out some of the main things that we discussed, hopefully to make it a little more clear for you guys, because it does sound a little confusing, everything together, but the mode of delivery plays a vital role in modulating colostrum microbiota. So C-section versus vaginal delivery, there's a big difference. The abundance of bacterial genera differed significantly between the two groups. Not the diversity, but the abundance, right? And so vaginal delivery was significantly richer in microorganisms belonging to Streptococcus and Haemophilus. Both kinds of colostrum samples analyzed in the present study contained a high percentage of intestinal microorganisms. Microorganismal interactions, or the microbiota network that we saw through AutoCM, they were significantly different between C-section and vaginal delivery colostrum. And the AutoCM data suggests that it's important to recognize that the pathogenicity of a microorganism is not always associated with its abundance in the environment, right? So just because you have more of something doesn't mean that another microorganism can't have a more significant effect on the overall interactions and relationships, but rather it involves several factors, including interactions with other microbes. So for the conclusion, I just have some take-home messages that I'd like to give you guys as I close out this presentation. So the mode of delivery had a large impact on the microbiota composition of colostrum. Further studies are definitely needed to better define the meaning of the differences that were observed between the C-section and vaginal delivery colostrum. When we were talking about the AutoCM map, we saw that only 5 hubs, those hubs being Acromobacter, Rhodanobacter, Ruminococcus, Serratia, and Staphylococcus, were shared between the two colostrum microbiota networks. The fact that they were shared I think it's a very important result of the AutoCM, and this means that they should probably be studied further. Finally, here are some questions for discussion. So question one being, what specific bacteria taxa were found to differ significantly between vaginal and cesarean deliveries in the colostrum microbiota? And what might these differences suggest about the transmission of microbial communities from mother to infant? Question two, how do the findings of this study compare with previous research on the impact of delivery mode on the neonatal microbiome, and what new insights does this study provide? Question three, what are the potential long-term effects of the observed differences in colostrum microbiota on the infant's immune system development and susceptibility to infections or allergies? Number four, in what ways could the study's findings inform clinical practices or interventions aimed at mitigating potential negative impacts of C-section delivery on the colostrum microbiota? And question five, last question, how might future research build on the results of this study to further explore the relationship between colostrum microbiota, delivery mode, and broader health outcomes in both infants and mothers? And with that, thank you guys for watching my presentation. And if you have any questions, whether it be for the auto CM or whatnot, just feel free to put it in the journal club and I'll try to answer them.